Assalamu alaikum. Today I will show you how PCR reaction is made and PCR is done and how primers are designed. A brief overview of yesterday's topic where we isolated DNA. Although we have isolated the DNA, sample 1 and sample 2 from two different samples, but to detect either the gene, specific gene in this sample or in this sample is wild type or mutant, we need quite higher copy number of that gene, which is not here in this isolated DNA. So we have to amplify that specific gene so that we will be able to detect either that gene is wild type or mutant. And for that reason we are doing PCR, which is a technique to amplify the specific gene. For that, first of all, we have to make a master mix. This master mix is composed of tag buffer. This buffer contains, either we can order it through company, but we can manually make it by combining trace hydrochloric acid, potassium chloride, ammonium sulfate, BSA, and some detergent like Triton X100 or Twin 20. We will combine different concentration of these chemicals and we can make this buffer for the PCR by ourselves. We have to add magnesium chloride to that master mix to have a stabilized reaction, and magnesium chloride in this reaction will act like something like cofactor for the polymerase. We have to add DNTPs because we have to amplify the DNA. For that we need adenine, guanine, cytosine, and thymine DNTPs. And then we need forward and reverse primers for the amplification, and I will show you in the next slides that how we will design our primers for a specific gene. And we need tag polymerase. It is a stable polymerase, thermostable polymerase, which has been extracted from bacteria, and it is quite thermostable polymerase. The quantities are a rough quantity I have mentioned here is like we will take tag buffer as 3x. 3x mean if we have one sample, then we will take 3 microliter of this tag buffer. If we have two samples, for example in this case we have two samples, we will take 3 multiplied by 2 tag buffer in this tube, and then we will add 1.2x of magnesium chloride. As we have two samples, then it will be 1.2 multiplied by 2 microliter of magnesium chloride. We can use 0.15 micromolar to 1 micromolar magnesium chloride, and it is quantities like 1.2x. We can add 0.3x DNTPs. As we have two samples, we will add 0.6 microliter of DNTPs, and we will add primers, forward and reverse primers like 0.6 microliter for one sample. As we have two, then we will add 1.2 microliter in this tube, and then at the end we have to add tag polymerase enzyme, which is like 0.1x. We can add like 0.2 microliter tag polymerase enzyme, and then we have to bring our total chemical reaction for one sample up to 15 microliter, as we have two samples. So the additional adjustment of volume is made by adding water. We have to add 9.2x water for each reaction. So for these two samples we will add 18.4 microliter water. Now we have about 30 microliter combination of these chemicals in this tube. We will vortex it, and it is our master mix. After that we will take 15 microliter of this master mix in this tube and 15 microliter in this tube. And after that, we will add 1 microliter of DNA from this sample to this tube and 1 microliter of DNA from this tube to this tube. Now we have the DNA in these tubes, as well as we have this chemicals which we will use, which we are going to use for the emulsification of DNA. After that, we will mix these tubes and we will run in the PCR. So first of all, we will close these tubes, and then we have this PCR. We will put these tubes in this PCR machine, and we will adjust the temperature. As you know that we have 3 temperatures: denaturation temperature, which will denature or unwind, unzip the double-stranded DNA. For that, we need 94 degrees centigrade temperature for 30 seconds. But before that, I will suggest you as an initial denaturation of the DNA, so that all of the DNA should be denatured, which we have put there for 5 minutes at 94 degrees centigrade. Then after that, to adjust the repeated cycles to 94 degrees centigrade for 30 seconds. After 30 seconds, the annealing temperature, where the primers will be attached with the unwinded DNA for the gene which I am doing PCR, is like 58 degrees centigrade. It is for 30 seconds. I will run it in this temperature, and then after that I will increase the temperature to 72 degrees centigrade for the elongation, so that the DNTPs I have put there should combine with the single stranded DNA molecule to elongate it. So within 1.5 minutes from single copy of DNA, we will be able to get 2 copies of DNA. And after that, as a 50th step, we can add like 72 degrees centigrade temperature for 5 minutes, so that all of the DNA which were there should be like double-stranded. So in this way, these 3 cycles which I have mentioned here, cycle 1, cycle 2 and cycle 3, these will be cycles like 30 to 40 cycles. We will adjust it in this way, but the initial 94 degrees centigrade temperature for 5 minutes and the final 72 degrees centigrade for 5 minutes, they will be only one time. And in this way, within 2 hours, we will get about 34.4 billion times quantity of the initial quantity of the specific gene which was there. For example, if we are emulsifying the SP7, if initially it has only 1 copy number there, after 2 hours, 3 hours, by doing this PCR, we will get 34 billion copy numbers of that SP7 gene. So that now it is a very big quantity in that small PCR tube, and we will be easily able to detect that gene in the gel when we will run it. That is the idea behind doing PCR. Now, the only puzzling thing in doing polymerase chain reaction is designing your primers. Sometimes the primers are not designed properly, and in this way the chemical reaction will not take place, or in other words, we can say that the DNA will not be emulsified. So for primer designing, we have this website. We have many websites for designing primers, but this website is like NCBI website. It is a website by National Institute of Health, a very reputed organization, and we can use this website for designing our primers. So when we will click in this website, this tab will open, and here we have this step, all databases, we have to just click here, and when we will click here, it will show all the databases, but we need this database, gene database, so we have to select it. And when we will select it, then this tab will open, and now we have our gene, and we can put this name of that gene here. For example, in our case, that gene is sp7 gene. I will write my gene name there. This is an example. I am making primers for sp7 gene. So if I will put I will write sp7 here and I will search it, then all the information about the sp7 genes in different animals will be available. For example, in this case, when we will click on this search button, then we can get like sp7 reference transcripts of messenger RNAs. We can also get protein reference, and we can also get a reference sequence for the gene, which is the DNA. So obviously we are making primers for the gene. We will click on the reference sequence gene. So if we will click on the reference sequence gene, then this tab will open, and in this tab here we have this tab for the gene. When we will click on the gene, then a complete sequence of this gene will be open. I have already opened it, and it will be like in this way. So this is the complete sequence of the gene, which we have taken from the NCBI website by the method I already showed you. So this is the sequence of the sp7 gene, which I have extracted from the NCBI website. Now I am going to make a primer for this gene. So it depends on me what implicon size I am selecting based on my study. For example, if I am interested to amplify the whole of the gene, my forward primers will be here and my reverse primers will be here. Now it depends again that what number of nucleotides I am selecting as a forward primers or reverse primers. We can design our primers manually, as well as there are tools which are like optimized tools online available, and databases which are doing these primer designs for us. For example, one of such websites which is very reputed for the designing primer is NCBI Primer Blast tool. So if we will open this tool, we can design our primers here by this tool. It will design primers for us. So for that, it's a very simple tool. We have to like open it by going through here, and then we have to put our sequence in this website, and it will design a lot of primers options for us. Now we can use, like, for example, for this gene, for the 888 implicon size, this website has made these primers for me. I can use them as a forward and reverse primers. What I will do is just I will get this sequence and I will order them through a company. There are a lot of companies available in every country which are delivering or which are making these primers, and they can like deliver us within 5 to 10 days. Even in some countries like within 1 day. So when we will design these primers, we will order them through companies, and they are quite like not expensive. They are we can do it. Everyone can do it. So when these primers are delivered, then we can like do our PCR reaction. Beside the NCBI optimized primer maker which is there, and we frequently use it. We have other options like this IDT option. We have these tools in IDT and this oligoanalyzers tool. We can make our own primers by going through these websites, instead of like we can like log into this website and then we can make our own primers. Biolabs, New England Biolab is another best option for us. We can use, it has quite good tools, primer designing tools. We can use this tool for making different type of primers. But just for the wild type primers making, we can use the primer blast of the NCBI. These are some other options, we can use them. While making primers, we have to be quite cautious, because there are some things which matters during designing primers. One of them is like it should be minimum of 18 base pair lungs, which will reduce the probability of second site hybridization on the DNA template. So if we will use less than 15 base forward or reverse primers, there are chances of unspecific binding, which will like destroy the purpose. That is why at least we should have 80 to 20 base pair lungs forward and reverse primers. Beside that, there is the TM, which should be like 50 to 60. The temperature should be between 50 to 60, around that. That is the optimum TM for the primers. For example, in our primers which I have made, the TM is like 59 to 60, and it's fine. And another consideration we have to make is that the TM for the forward and reverse primer, the difference should not be more than 2 or 3. It's like 59.9 and the other one is 60, so it's a very ideal situation where the TM for the forward and reverse primers are same. When the difference between TM for the forward and reverse primer exceed 2 or 3 degrees centigrade, then it will not help us properly. And then another consideration is the GC content, the guanine cytosine content in our primers. They should not exceed 60. Between 50 to 60 is like optimum. For example, in this primer it has 50 GC content, it has 60, it has 50 50 50 50. That's fine. But if the GC content increases beyond 60, it will change the TM of the primers, and then there are certain issues like self-complementarity, or primer dimers are formed due to these problems. Some other issues which are there, and we have to give proper consideration to them while designing our primers, are the GC content. As I mentioned, we have to make it like 50 to 55 to 60 or 50 to 55. We should not increase it, and then ensure the primer is as pure as possible. If there are some contamination, then we cannot get our desired product. And then sometimes the primers that can hybridize to from primer dimers should be avoided. Means the primers sometimes they make a circular structure, and instead of attaching with the DNA templates, they starts circularization, or they start unspecific binding with each other. These are the issues which we have to avoid. And then if there are certain palindromes, that may be issue. So while designing primers manually, we have to give due consideration to these aspects. And that was all for today. We discussed the PCR, and we discussed how the how the primers are made. These are some basics of the PCR and primer making, and in next video I will tell you that when we amplify the DNA with by the PCR, then after that how we can run it in the gel to see whether a specific gene is mutant or wild type. Thank you so much.